Hello, my name is Agnes Koerfer and I'm a PhD student of Christian Eggeling's group in Jena, and I want to give you a short introduction in the idea of my project. One of the main interests in Christian's group is to understand cellular membrane organization by studying the diffusion of its components. For this, we combine super resolution microscopy techniques with fluorescence correlation spectroscopy. And we are setting up a simulation toolbox to understand theoretical diffusion models better and help us interpreting experimental data. The easiest assumption of the dynamics of the membrane molecule is to describe them with Brownian diffusion. Here, the particle does a random walk over time with a defined diffusion coefficient. This behavior was shown to be the case for PE in the plasma membrane, for example. But nature is more complex than this, and it was shown often that pure Brownian diffusion is not sufficient to describe all the dynamics in cell membranes. The main diffusion model or behavior I'm interested in is the so-called trap diffusion, where the particle stops its movement kind of randomly in a spot or small area due to interactions with the partner or its environment. This is called a trapping event, and the behavior was first described for single myelin in the plasma membrane. The reason and in our simulation, we implemented this behavior by introducing trapping sites where the particle can enter and leave with a certain probability, resulting in an average trapping time, the time the particle stays in such a trapping site. The reason and the dynamics of those trapping events so far are not fully understood, and to maybe get one step closer to this is one of our goals. As I said before, our main technique is fluorescence correlation spectroscopy, where the diffusion of fluorescent particles is studied by measuring the intensity in an observation spot over time. The collected signal is correlated to extract the average transit time and therefore being able to calculate the diffusion coefficient. The advantage of this technique is that it has single molecule sensitivity, but giving an average results to remain good statistics, and also being able to achieve fast sampling rates. To distinguish between different diffusion modes, the size of the observation spot is varied, and the according diffusion coefficient is calculated. For Brownian diffusion, the diffusion coefficient is independent of the observation spot size. For trapping behavior, due to the longer transit times induced by the trapped particles, the diffusion coefficient is expected to be much smaller for smaller observation spot sizes. By increasing the size, the trapping time loses more and more the prone influence on the transit time, leading to an increasing behavior of the diffusion coefficient. To test our implemented diffusion models, for the simulation, we defined a simulation area where the tracks for numerous particles were calculated with a certain diffusion coefficient, and later an FCS measurement was simulated. By varying the spot size, we could reproduce the constant behavior for Brownian diffusion. For trap diffusion, trapping sites were introduced and the simulation was repeated, leading to the expected increasing behavior. But we don't want to stay only in one point, but extend it to a whole image stack. To not get temporal information in only one point, but additionally in a whole area, as well as the spatial information, to hopefully understand the dynamics of those trapping events a little bit better. I want to thank my supervisor, Christian Eggeling, and my colleagues for the interesting and fun work. And if you have questions, feel free to contact me.